Hi guys, I'm Tobias,
And I'm Selihom.
and we're here to talk to you about sex-linked traits and gene inheritance. First, let's do a brief recap of some basic genetic concepts. Genes are encoded in our DNA. Our DNA is packaged up into chromosomes. Those chromosomes are passed on from parent to offspring. Now, us humans have 23 chromosome pairs, the last being sex chromosomes, X and Y. One of the most basic concepts of inheritance is that we receive one copy of each chromosome from our mother and one from our father. You'll notice that for the sex chromosomes, the one from the father looks different. This is because Y chromosomes are shorter and contain less genetic information, which is why X-linked genes are more common than Y-linked genes. Loci on autosomes follow a Mendelian pattern of inheritance, where genes follow the laws of segregation and independent assortment. Sex-linked genes, however, follow a sex-linked pattern of inheritance because loci lie on sex chromosomes. So we find that one sex may be more likely to display a mutant phenotype based on which parent contains a given mutation. For the purposes of this video, we will be demonstrating sex linkage by primarily looking at X-linked recessive mutations and how the inheritance patterns differ from autosomal recessive mutations.
For this autosomal cross, we'll start out with two parents. You'll notice that we haven't indicated which is the mother or the father. This is because it doesn't matter for autosomal mutations. We'll be looking at an autosomal mutation located at what we'll call gene A, of which one parent will be homozygous recessive, denoted as A-, and the other will be homozygous dominant, which is A+. You can see on this first Punnett square the expected genotypic frequency of the offspring. All offspring would end up being heterozygous with the wild-type phenotype. In the second cross, we'll determine the expected genotypic frequency of offspring when the alleles are sex-linked. The female parent has two X chromosomes, meaning that she'll carry two alleles for the gene in question. In this case, both of those alleles are recessive, making her homozygous recessive. The male only carries one X chromosome that is wild-type for the gene in question. For this cross, the Punnett square shows that all offspring who are male will carry this recessive allele since they receive their single X chromosome from the mother, whereas all offspring who are female will be heterozygous and phenotypic all wild-type since they receive one mutant copy from the mother and one wild-type from the dad. This third cross is a reciprocal cross of the previous one, and will determine that the expected genotypic frequency of offspring for a sex-linked trait when the male parent carries the mutant allele on his one X chromosome and the female parent carries two wild-type alleles on her two X chromosomes. The Punnett square shows us that all offspring, regardless of their sex, will be phenotypically wild-type. So, we see that unlike autosomal mutations, sex-linked mutations have different genotypic and phenotypic ratios based on the sex of the offspring. Understanding these different ratios allows us to determine sex linkage. So, what's one way for us to study sex-linked alleles?
Fruit flies. We'd be looking specifically at a gene affecting eye color to demonstrate sex linkage. The white gene, which is located on the X chromosome of fruit flies, is a sex-linked gene. The wild-type phenotype for eye color in flies is red. However, mutation in the white gene will result in a fruit fly with white eyes, which is the mutant phenotype. In this first cross, we will determine the expected genotypic frequencies of progeny from a cross between a mutant female and a wild-type male. We see that the male parent is wild-type for red eyes, and the wild-type allele, which we will denote as W+, is on his X chromosome. We also see that the female parent carries two mutant alleles, which are each denoted as W-, meaning that she is phenotypically white-eyed. In our offspring, we see that all male progeny inherit the W- allele on their X chromosome, and all female progeny inherit both the W+ and W- alleles on each of their X chromosomes, making them heterozygous. This means that all male progeny will be phenotypically white-eyed because they only inherit the mutant allele from the female parent, while all female progeny are heterozygous wild-type. The second cross is a reciprocal cross of the first slide. Here, we will determine the expected genotypic frequencies of a progeny from a cross between a wild-type female and a mutant male. In our progeny, we see that the female progeny inherit one mutant allele from their father and one wild-type allele from their mother, and the male progeny all carry the wild-type allele inherited from their mother. Thus, all female progeny will be heterozygous wild-type, and all male progeny will be wild-type. This means that all progeny will be phenotypically red-eyed.
So, fruit flies are cool, but why does this matter? Understanding how sex linkage works is valuable in the field of genetics because sex linkage causes many conditions you've probably heard of, such as hemophilia, male pattern baldness, and Fragile X syndrome. So, Tobias, what did we learn?
First, we learned that sex-linked genes do not follow a Mendelian pattern of inheritance, whereas autosomal genes do.
Second, we learned that if we want to determine if a gene is sex-linked, conducting phenotypic analyses on model organisms such as fruit flies can help us do this.
Lastly, we learned that sex linkage affects organisms with distinct sex chromosomes, including us. That's all. Thank you for listening.